Good afternoon. It's a pleasure to be here with you in Angers and discuss some of our work on the epidemiology of cavity spot, which is a very important disease in California. California produces most of the carrots in the United States. Cavity spot's probably the second most important disease after the root knot nematode. Okay, so what species of Pythium cause cavity spot in California? It's primarily Pythium violi and Pythium sulcatum. And we do a survey every year, not because those organisms change in importance, but because we're concerned about the development of resistance to fungicides. But we haven't seen any resistance build up in any of our species, but there is one that we call Pythium neo-irregulare. That's not very common, but is completely insensitive to ridamil, to methoxanum. So we always keep our eye on this one, but so far it's in our soils at some low level. And it didn't develop resistance in response to our applications of fungicides. It was always resistant, because we can find this in fields that have never had an application of methoxanum. So Pythium violi and Pythium sulcatum are our primary pathogens. And so in our first trial, we wanted to know the effects of different crops on the incidence of cavity spot and Pythium activity. So we grew carrots following carrots, carrots following onions, barley, alfalfa, and cotton, and so forth. And in this experiment, we grew the crops, we incorporated the crop residue, and then planted carrots within a week after incorporating the residue. We did that because we wanted to know the effects of the residue on Pythium. And so a couple of these crops, barley and alfalfa, increased the CFUs, the colony-forming units of Pythium in the soil. So Pythium, it turns out, is a fast colonizer of organic matter. If you incorporate crop residue, there's a succession of organisms. Within 24 hours, the zygomycetes grow very quickly. Soon after that, it's a species of Pythium. Following that, in a week or two, it's many ascomycetes, like trichoderma, for example. And then the tougher residue, like lignin, are colonized by the basidiomycetes. So there's a distinct succession of organisms. Okay, so our next experiment included 3 treatments: an organic treatment where the nitrogen was just supplied by green manure, incorporating green residue; a treatment we called transitional, which was mostly animal manure, composted animal manure; and then a conventional treatment that included a treatment with metam, or vapam, metam sodium. And that's pretty common use in California. So the experiment went cover crop in the winter, or nothing, or fallow, then a spring crop of potatoes, another green manure crop, cover crop in the summer, in this case black-eyed beans, which also is a nitrogen fixer, more animal manure, and then in that conventional treatment, an application of vapam. Then we planted carrots in the fall. And this was repeated in the same plots in the same field every year for 3 years. And we periodically took samples, but just in summary, generally, the number of fungi in the soil samples from the organic fields, the fields that had the incorporation of the green manures, was significantly greater than the other plots. That was also true with bacteria in general. And then we also measured microbial activity with a FDA test, at fluorescein diacetate, which causes a color change in response to enzymes produced by all organisms. So a color change can be quantified, and you get an indication of the microbial activity. And as expected, there was more microbial activity where we incorporated the green manure. Okay, we also looked at pseudomonads, which is often an indicator of soil health and organic matter and microbial activity, and as expected, more pseudomonads in the organic treatment. The Pythium populations are often higher, because as soon as we incorporated that green manure, the Pythium populations went up. You know, that's not a good thing, but later they went down. And then nematodes, we had some mixed results with nematodes. And then the yields in this trial were generally higher in the conventional treatment, which received inorganic sources of nitrogen fertilizers, which is not unexpected, because it would take some time, some years, for the nitrogen to become fully available from the organic sources. Okay, so to summarize this experiment, this is disease incidence, and right after the incorporation of green manure, we actually saw more disease caused by Pythium, we're working with Pythium here, than the other treatments. But that was only within 10 days or so, about a week. Following that, in the second and into the third week, we saw disease levels go down. So our soils became suppressive. But the problem is only briefly. We lost that suppressiveness by the third or fourth week. So it didn't last long. Okay. So that led us into the next experiment, where we thought that the green manures that we were using previously, sorry, I didn't mention, were vetch and peas and an oat mixture. And these were very herbaceous, obviously, and we turned them under, and the sugars became available immediately. So in this next experiment, we used some more woody sources of manure, green waste, we call it. So one treatment is called green waste, and so it's not fully composted. It's still biologically active. Another treatment received composted green manure. So this is cold. It's not active any further. And then a treatment we called fallow, and then the conventional. And the differences were in the green waste, we incorporated this green waste material, spring crop of potatoes, more green manure in the summer, and then carrots in the fall. The next experiment, the fully composted green manure, added in the winter and summer, carrots in the fall, fallow 2 periods, and then in the conventional, there was an application of VAPAM, metam sodium. Okay, so here's just a picture of these different kinds of green waste. So this one is the composted green manure, and this one's not fully composted. You can kind of get an idea. It's also woodier, so there's a lot more hemicellulose and lignin in this compost. So our idea is that it's not going to break down as fast. We sustain levels of organisms in the soil, increase microbial activity, and therefore prolong the disease suppressiveness of the soil. And to summarize some of this data, both with the green waste and the compost, we see elevated numbers of fungi and total bacteria and microbial activity. We do this periodically, so I have a number of data points. But you can also see that it fluctuates greatly. So we're able to increase the microbial activity here. This is that FDA value. But sometimes the beneficial effects drop off rather dramatically. So sustaining this benefit is the challenge. This is last year's data. Again, maybe this one shows it more clearly. So that green manure was incorporated in the beginning of March. And so you can see the spike of microbial activity here in March. But you see by May, it's essentially gone. At least in the green material, it was gone. So it was sustained a little bit longer with the composted material. By July, it's completely gone. And our soils are warm. You know, in California, the soils are quite warm. Okay, so how do we measure disease? Well, although we're studying cavity spot is a disease that can occur early in the season, but generally occurs in the last half of the season. So we developed an assay that substituted for that, where we took soil samples from these various plots, and they're all replicated 4 or 5 times. And each plot we took soil samples back, we brought them back to the lab, and we spiked the soil samples with Pythium. And then, because we wanted a rapid turnover, we planted cucumber seeds, an exact number of cucumber seeds. And so then all we had to do was count the number of surviving cucumber seedlings to get an idea of the disease suppressiveness of the soil. So the higher the value here, the greater the disease suppressiveness of the soil. And you can see we really increased seedling survival in the soil samples that were amended with the green manure, waste, and compost. But it does jump around. It does drop off. Sometimes there's a spike with the conventional. That's because we applied VAPAM back in September. So actually, that reduced Pythium and Rhizoctonia and everything else in the soil. This is again an indication of disease suppressiveness last year. And again, you get the same idea. So we have some spikes here of disease suppressiveness, but they don't last long. It doesn't last long. There's still some here with the compost, but with that green waste, it drops right off. Okay, so there is a relationship between microbial activity and seedling survival. That's a pretty strong relationship, that top figure. There was also a strong relationship between organic matter and seedling survival. So it's their organic matter that we're trying to elevate and get these benefits. But it's difficult to do in our soils in California. These are calcareous soils. They're very, very warm. We add organic matter, and it decomposes, it combusts very quickly, as we've seen. So it's very difficult to increase the amount of organic matter in our soils. That's really our challenge. Now, in 2014, I just got the results back, the green manure and the composted manure were between 2 and 3% now. So that's really significant for us. But still, it's very difficult to do. The organic matter decomposes very quickly. Okay, so we furthered this by looking at the total community profiles in the soil. So this is a high-sequence tool to look at bacteria and fungi in the soil. And I won't talk about the details of this. Some of you know Illumina sequencing and 454 sequencing. But there was a significant increase in the diversity of bacteria where we applied the compost. And this is what we think is providing our disease suppressiveness. And this just gives you an idea of all the different groups. We have tens of thousands of data points, and you try to tease it apart, and we couldn't get, we know we increased the diversity. You can just kind of look at the colors there, but we didn't increase any one organism. And we tried to tease it apart, but there are no significant differences in the populations of bacillus or pseudomonads or streptomyces, groups of organisms that you often attribute to disease suppressiveness. We looked at fungi, but there was no significant difference in the community profile among fungi. And finally, we also did this community profiling in 4 conventional fields. On the top, these are just names of areas in California, but 4 conventional fields. This is where they'd use inorganic sources of nitrogen. Organic matter wouldn't be applied. VAPAM would be applied, and it had high levels of cavity spot. In organic fields, we chose 3 organic fields with low levels of cavity spot, and we saw significantly greater bacterial diversity in the organic fields. So finally, okay, I'm done. All right, so conclusions. Organic matter encourages microbial activity, which creates a disease-suppressive soil. And we couldn't identify a single organism, so we think it's by the community. But unfortunately, the organic matter decomposed very quickly for us. Our temperatures are very warm, and so it's short-lived. Thank you.
Thank you. The soil becomes suppressive to PCM. Is it suppressive to biological control agent like trichoderma? Is it possible to use biological control agent in this soil?
Yeah, so we tried biological control agents. We have tried individual agents like trichoderma and other materials, but we have not been able to get them established. So we have introduced them to the soil, but their activity is very short-lived. So that's been a problem for us. We have not been successful. Maybe if we apply, for example, trichoderma many times, but yeah, we haven't been successful.